So let's remember something. Meiosis reduces the genome from diploid to haploid. That means from an adult with 2 homologous chromosomes for every chromosome to haploid, which is a single chromosome. So if we were to start at, let's say, fertilization, we would see we go from fertilization, and that restores a diploid zygote. Then, all along here is mitosis and develop. So during development, you have mitosis going on. First, for all the cells, until you finally get a baby, and then there's continuing development until sexual maturity. At that point, we have a male and a female. They have sex. Well, hold on a second. They don't have sex yet. They each produce, by meiosis, haploid daughter cells. So, haploid gametes, N equals 23, and sperm and egg. Okay? And then, of course, we're back to fertilization until we have a restoration of the diploid zygote. Okay? But one thing I forgot to mention earlier in regular lecture is that once you have a diploid zygote, after that it has to divide and develop, and so the only way to do that is through mitosis. This is an egg with many sperm trying to fertilize. Let's just remember, sex is usually very costly, or it can be. Large amounts of energy required to find a mate. Intimate contact, so perhaps STDs, genetic costs. With asexual reproduction, we pass on all our genes to our identical clones. With sex, we only pass on half. And then males actually are a luxury. They're just a sperm delivery device. And oftentimes they contribute very little to rearing offspring. But that's not exactly true in humans. But okay, more genetic diversity, a lot more. So more potential for survival of genetically diverse individuals when environmental conditions change. So you have a changing condition, you have these individuals that are very different from each other. There's no two alike unless they're identical twins. And some of those genotypes and phenotypes might be well adapted to newer environmental conditions compared to the rest of the population. Then they, of course, will be well adapted. They will pass on more of their genes to future generations. And so what happens is you shuffle of genes in meiosis, crossing over, random fertilization, and then there's this whole idea that meiosis might be a part of DNA backup and repair. Asexual organisms don't have backup copies of genes. Sexual organisms have 2 sets of chromosomes. So one can act as a backup to rescue gene expression if there's damage to one. Sexual mechanisms, especially recombination, are used to repair damaged DNA. The undamaged chromosome acts as a template, and eventually both chromosomes end up with the correct gene. So each chromosome of a homologous pair comes from a different parent, and each chromosome might have different alleles at corresponding loci. So a quick look at meiosis. So meiosis 1, of course, you still have interphase, so you're doubling the chromosome. Then in prophase, there's this idea of crossing over. We'll get into more specifics. Metaphase 1 and anaphase 1. And so let's look at this process. Maternal and paternal chromatids and homologous chromosomes exchange genetic information through crossing over. We'll look at that shortly. Metaphase 1. Independent assortment results in random combination of maternal and paternal chromosomes in the gametes. And then anaphase and then telophase and cytokinesis, homologous pairs of chromosomes then separate, producing two daughter cells. Okay. Genetic recombination by crossing over further increases genetic variability. The exchange of corresponding segments between two homologous chromosomes. So, if we actually look at this, we see right here, for example, an area where they're going to switch and exchange. And then, of course, you can see what's happened now. Instead of this still being part of large C, it's now part of small c. And here it is again. So this happens during prophase 1 of meiosis. So genetic recombination results from crossing over during prophase 1. So in other words, this is really important in terms of the benefits of sexual reproduction and why each gamete that is finally produced is actually unique from both parents and other offspring. Okay. Then there's also independent assortment. So as you can see, we've got a homologous pair of blue chromosomes, homologous pair of red chromosomes, one from each mother, right? These are now called tetrads. And in this case, we only have 2 chromosomes. And they can be pulled apart from metaphase to anaphase from metaphase to anaphase to different sides of the cell. Okay, now at the same time, so that was this, right? So the red goes this way, the blue goes this way. Okay, now at the same time, chromosomes can actually line up so that in the first possibility they're X from the father, Y from the mother. In this one, you can have X from the father, Y from the mother, but on the bottom, instead of having X from the father, Y from the mother, you now have Y from the mother, X to the father. Okay? And so you can see these blue chromosomes are both from the father, and then both from the mother, but because of this crossing over that happened, the same thing, metaphase, father, mother, but you can also have mother and father. So, sister chromatids of each chromosome separate, result in 4 haploid daughter cells. Okay, so we're going from homologous to haploid, or diploid to haploid. And then metaphase, there's more independent assortment, like we saw in metaphase 1. Okay, and between metaphase 2 and anaphase 2, these homologous chromosomes can then pull apart and go in either direction. So here we are again, metaphase 2, we finally end up in telophase, where we have perhaps a small red chromosome and a large red chromosome, or a small red chromosome and a mixed chromosome of dad, mom, dad, or a small blue chromosome of mom, dad, mom, or two blue chromosomes in the haploids. So you can see the variability.